and this is actually a proceedings presentation, so there was a paper associated with this that was published in ISMB. It's about predicting CTCF-mediated chromatin loops using CTCF-MP.
Hello, everyone. I'm Ruochi Zhang, a first-year PhD student from Carnegie Mellon University, and I would like to thank the organizer for this great opportunity to present this work named Predicting CTCF-mediated chromatin loops using CTCF-MP. This is the outline of this presentation, and first I'll briefly introduce the biological motivation and background of this work. Three-dimensional organization of the chromosomes in the human genome is critically important for understand the principles of gene regulation and disease mechanisms. Recent high-throughput globin mapping methods such as Hi-C and ChapHat have revealed that higher-order chromatin architecture actually harbors much more complicated chromatin interactions than we previously know. As the figure in the right side of the slide shows, there is actually numerous chromatin interactions within one TAD. One of the most interesting examples involves the architectural protein CTCF. CTCF is known to have the capability of forming loops, which I'm sure you have heard a lot of times during this meeting. And also, both high coverage Hi-C and ChapHat has shown that most of the CTCF binding sites in the loop anchor region is actually in convergent orientation, as shown in the lower left side of these slides. This could indicate that the motif orientation actually plays a critical role in the loop forming process. However, there are still several important questions related to CTCF loops that remain unanswered. First, for CTCF protein that binds to certain CTCF binding site, it may not be able to form chromatin loops with other sites. Second, for a pair of convergent CTCF motifs that are both bound by CTCF protein, it also does not always form a chromatin loop. So what could be the factor that decide whether a pair of convergent CTCF motifs would form into loop or not? And more importantly, how much is the sequence level feature contributed in this process? Our goal is to answer those questions by using a machine learning algorithm to predict convergent CTCF loops. This image shows an overview of our model, CTCF-MP. We tackle this prediction problem by regarding it as a binary classification one. We first generate positive samples and negative samples from CTCF ChapHat data under a strict standard. Here, both positive and negative samples are pairs of motifs. Then we extracted features from CTCF motifs with its flanking region and also the window region between the motif pairs. We use a word-to-vec model to encode DNA sequences, and we also use additional features, includes branch of origin score, CTCF motif matching score, etc. After that, we trained a boosted tree classifier to predict whether a pair of motifs would form into loop or not. And finally, we evaluate the model using several metrics. For the data processing part, we use CTCF ChapHat data for 4 cell lines from both ENCODE project and from this work from town. For a certain cell line, we only consider CTCF motifs within CTCF trypsic peak region, which indicates that these motifs are actually bound with CTCF protein. We define positive samples as convergent CTCF motif pairs within ChapHat detected loop regions. We define negative samples as the rest of the convergent CTCF motif pairs. After that, we would sample the negative samples with similar distance distribution as positive ones. And we did this because it is known that the distance between a motif is crucial to decide whether they would form into loop or not. So we want to reduce the contribution of the distance feature so we can focus on other important factors as well. For the feature extraction part, we use word-to-vec to encode genomic sequences. Word-to-vec is a popular word embedding model in natural language processing field. It reduces the dimensionality of the words while keep important information such as the relationship between words. Here I'll briefly introduce the basic idea of this model. It's a 2-layer neural network that is trained to predict the target word from its context. As As this figure shows, for example, it would try to predict the word mat from the context the cat sits on the. And this image also shows an intuitive way to implement this model. However, this naive implementation is computationally very expensive because, as it shows, the last layer needs to do softmax over all the word in the vocabulary. So in Word2Vec, they use a trick called negative sampling, which means that instead of learning the full probabilistic model, it trained a binary classification instead. Now it would try to distinguish the target word from the noise word, so it would try to maximize the probability of the target word with its context being observed together, while minimize the noise word with the context being observed together. And here the noise word is generated from a noise distribution, which is usually the most commonly used word, like I, he, she, etc. And this model is much more computational efficient than the last one. After training this binary classifier, the parameters of the hidden layer could be regarded as the embedding vectors for words. And usually for a sentence, we would use the average embedding vectors of its words as the embedding vector for that sentence. To use Word2Vec here, we need to define what are sentences, words, vocabulary, some term like that in biological context. So as this table shows, here we define DNA word as subsequences of fixed length k, which is also referred to as k-mer. We define vocabulary as all possible k-mers. And if you scan a DNA sequence with fixed window size k and step size 1, you would get a DNA sentence. Word2Vec has previously been used for similar tasks before. For example, last year we developed a model using Word2Vec to encode genomic sequences and to predict promoter-enhancer interactions, and this work is published in bioinformatics. And there are other works that use Word2Vec to encode protein sequence, such as this one in PLOS1. Besides the Word2Vec encoded features, we use some additional features, including sequence-level features and functional genomic signals. Sequence-level features we use includes branch of origin score, which I'll introduce later, and distance between the motif pair, motif occurrence frequency in the window region, and also GC content for both CTCF motif with its flanking region and also the window region between motif pair. The functional genomic signals we use include CTCF trip-seq signals and DNA-seq signals. Now I'll introduce one of the most important feature we use here called branch of origin. This model is developed to quantify the TF binding site conservation by taking the turnover event into account. It assigns the emergence of a lineage-specific TF binding site in the human genome to a particular branch in the mammalian phylogeny. To understand in a much easier way for a specific TF binding site in human genome, we would want to know when is this TF binding site come into being at the very beginning. For example, in the evolutionary history. For example, like this binding site first appear in elephant or mouse or chimpanzee, something like that. This figure shows tracks of branch of origin score and loops detected by TriPET in GM cell line. Here, higher branch of origin score indicates earlier emergence, and we split the branch of origin score into branch of origin recent and branch of origin ancient based on whether the emergence is earlier than the primate common ancestor or not. As you can see, most of the loops are formed by CTCF motifs that emerge earlier than the primate common ancestor. And I think this finding could partly answer our first question that for individual CTCF binding site, what could be the reason that decide it would form loops with other sites or not? Using all the features mentioned above, we train the boosted tree classifier, and here's the performance evaluation. As you can see, CTCF-MP performs pretty well across all the cell lines. Note that here for all the experiments, they are done to individual cell line, meaning that the training set and the testing set are actually from the same cell line. And as mentioned above, we sample the negative samples to have similar distance distribution as positive ones, so it's a balanced data set. And we also tried CTCF, We also tried CTCF-MP on an imbalanced data set just to show the whole genome, just to show the performance on the whole genome data set. And here's the number. Still, CTCF-MP is really good at the whole genome data set as well. Here's an image illustrating a visualization of certain features used in the model and also the prediction decision made by CTCF-MP. The bottom 4 tracks are loops detected by CTCF-Chapter, the loops captured by our model, the false positive decisions made by the model, and the loops we missed in the model. Still, you can see there is not much false positive and false negative here. Then we asked, can we use CTCF-MP for cross-cell type prediction, meaning that we would train CTCF-MP using data of one cell type and test it on another cell type. To avoid the using testing samples in training samples scenario, here both training and testing samples are cell type specific motif pairs. And this figure shows the AUC of cross-cell type prediction. Even in the worst scenario, CTCF-MP can reach 0.75 AUC score. And similarly, we visualize the process of it. For this visualization, the model is trained by using GM cell lines data and tested on HeLa cell line. As the right pane here shows, the model trained by GM cell line captured a cell type specific loop in HeLa cell line, indicating that the features learned by the model is actually consistent across cell line. After proving that CTCF-MP is good at predicting loops, we want to further show that the feature learned by the model is actually biologically meaningful. We first show that the sequence features alone can have great predictive power. As this table shows, using sequence features only, we can get a comparable performance. And as expected, if you combine that with functional genomic signals, the performance would go higher. We also show that the important word-to-vec features are shared over cell lines. We estimated the feature importance by using the average information gain when certain feature is used in the decision tree. And the heat map plots the top 30 important features across cell line. As you can see, the most important word-to-vector feature is shared across cell line, indicating that the sequence level feature underlying the CTCF loops are actually consistent. To sum up, we developed CTCF-MP, an effective computational method that can accurately predict loops formed by convergent CTCF motifs bound by CTCF protein. And we can use CTCF-MP for cross-cell line prediction. Finally, our results offer important new insights in the sequence-based features underlying loop formation between a pair of CTCF motifs. Last but not least, I would like to thank everyone who helped me during this project, including my advisor Jen Ma, the co-authors Yu Chuan, Yang Yang, and Yang Zhang, and also everyone in the lab. And also I would like to thank the funding source. Thank you for your attention.
Very interesting. Do the loci that do come together have more CTCF binding sites than the loci that don't form loops?
Since CTCF-Chapter data is like specific to CTCF protein, so all the interactions detected by Chapter are actually, you can find some CTCF binding sites here. And we also checked for the interactions from high-C data, and I think, I don't remember the number, but yes, like most of the interaction has CTCF binding sites near that loci.
My question is about the feature importances. What are the words that you found that were predictive of the interactions?
So you want to maybe see like motif stuff
Just what
Something like that.
What were they? I'm curious.
Okay, usually here like we use K as 6, so they are like 6-mer. I can't actually remember like what they are, but I think one of the most interesting, or not that interesting thing, is that we found the most important sequence level feature is like the CTCF motif, the CTCF consensus motif. So it makes sense, but not that interesting, so we didn't include that here.
Beyond that?
Beyond that, sorry, I don't quite recall like in detail what are they, but thanks for your question.
Thank you for interesting talk. My question is, what is the advantage of using word-to-vec instead of one-hot vectors?
Instead of what?
One-hot vectors.
The one-hot encoding.
DeepC uses.
Okay, I think we compared the using word-to-vec and just the K-mer frequency, and word-to-vec outperforms using K-mer frequency. And the advantage of using...
But DeepC uses one-hot vectors.
Oh, one-hot vector.
Like 001 is A and 0010 is G.
Then the dimensionality for that would be pretty high, right?
No, I don't think so. Actually, DeepC works well, right?
Yeah,
So,
okay.
what have you compared?
Okay.
Hi, I have a question about the negative samples. You said you only consider CTCF motif within the CTCF chip-seq peaks. So is that the same case for the negative data set?
Yes, it's the same.
Okay, so why don't you try to consider for the negative data set, the CTCF motif outside the chip-seq data?
One of the reasons is that for the CTCF motifs that is not within the CTCF chip peak region, they usually does not form loops. So if we include that in the negative samples, the task would be like too easy.
Okay.
Okay. Okay. Thank you.
Let's thank our speaker one more time.